Hi everyone, my name is Alejandro López Amador from CIDETEC Mexico, and today I'm present our work entitled Molybdenum Disulfur NANOZYMES for Electrochemical Hydrogen Peroxide Sensing and Antibacterial Activity towards Oxidative Stress-Mediated Nanobiomedical and Applications. In this work, we explored molybdenum disulfur or NANOZYME with peroxide-like activity, focusing on the potential for antibacterial application and electrochemical hydrogen peroxide sensing. To provide context, it is necessary to say the antibacterial or antimicrobial resistance is currently a major global health problem, creating an urgent need for alternative antimicrobial strategies. One promised approach is the use of nanozymes, which are nanomaterial capable of mimetic catalytic activity of natural enzymes. In particular, molybdenum disulfur is very interesting because its physicochemical properties can be tuned by controlling in the structure, morphology, and effect density. In this study, we focus in the peroxidase-like activity. This catalytic activity can promote the conversion of hydrogen peroxide into high reactive oxygen species. These species can interact with the bacterial cells and producing oxidative damage to membrane and the other cellular compounds. Therefore, our main object is to investigate the relationship between the physicochemical properties, peroxidase activity, antibacterial performance, and electrochemical detection of hydrogen peroxide using molybdenum disulfur-based nanozymes. As I show in the methodology section, the molybdenum disulfur nanozymes were synthesized using hydrothermal method at 200 degrees Celsius for 24 hours. We prepare material using different molar ratios of molybdenum and sulfur precursors, particularly 1:1 and 1:4 ratios in the order to modify their physicochemical and catalytic properties. The resulting materials were characterized use FTIR spectroscopy, Raman spectroscopy, scanning electron microscopy, and transmission electron microscopy. Next, their peroxidase activity was evaluated using TMB-SI. In this reaction, molybdenum disulfur catalyzed the reaction between hydrogen peroxide and TMB, producing oxidative TMB, which the characteristic blue color and absorption band around 615 nanometers. We also evaluate the antibacterial against two bacterial models, Staphylococcus aureus as a gram-positive bacterium and Pseudomonas aeruginosa as a gram-negative bacterium. Finally, hydrogen peroxide was evaluated electrochemical using a platinum working electrode and rust production in additional investigation using fluorescent probes. Moving to results, the physicochemical characterization confirmed the successful formation of nanostructure molybdenum disulfur. The X-ray diffraction patterns show the characteristic repletion associated with the molybdenum disulfur, while FTR spectroscopy showed vibration relate the molybdenum sulfur bonds. More important, the Raman spectrum showed the characteristic E2G1 and A1G vibration models of the molybdenum disulfur. Difference between the samples can also be observed, particularly in the low wave number regions, suggesting structural modification associated with the different synthesis conditions. The microscopy images also demonstrate the changing the molybdenum to sulfur ratio significantly modifies the morphology and organization of the resulting nanostructure. These structural differences are particularly important because the catalysis properties of the molybdenum disulfur are strongly influenced by the species, age, defect, and accessible activity sites. We investigate um, we investigate the peroxidase activity at the NANOZYME. As shown in the kinetic curves, an oxidation of TMB strongly depends the concentration of the molybdenum disulfur nanozymes. As the amount the nanozyme increment, we observe progressive higher absorber indicate increased formation of oxidation TMB. We also evaluate effect TMB concentration and against observer concentration depend catalytic response. The results confirm that our molybdenum disulfur materials exhibit intrinsic peroxidase-like behavior in the presence of hydrogen peroxide. We additional performance kinetic analysis to obtain parameters such apparent Michaelis-Menten constant and maximum reaction velocity. Interestingly, difference we observed between two 1 to 1 and 1 to 4 molybdenum disulfur samples. This suggests that modifying the precursor radium during the synthesis can influence the catalytic properties and resulting nanozymes probably through change in the structure morphology and availability catalytic sites. In the next part, we evaluate where this catalytic behavior could associate with the antibacterial activity. The nanozymes were testing against Staphylococcus aureus and Pseudomonas aeruginosa using controlled hydrogen peroxide conditional. As shown in the graphs, both molybdenum disulfur formulation exhibit significant antibacterial activity. For Staphylococcus aureus, the treatments production very high antibacterial activity after both 3 and 24 hours. A similar behavior observed for Pseudomonas aeruginosa, although some difference between the formulation and concentration we observed. Importantly, control experiments with the hydrogen peroxide along with performance to distinguish in the intrinsic effect the hydrogen peroxide from the catalytic contribution on the nanozymes. Overall, the results support the hypothesis that the antibacterial performance is enhanced by catalytic activity of the molybdenum disulfur in the present hydrogen peroxide, rather that begin explained only by hydrogen peroxide itself. Another important part of this work was electrochemical detection hydrogen peroxide. We used three electrochemical system with a platinum working electrode. First, we obtained the calibration curve for hydrogen peroxide and micromolar range. As shown here, in very good linear relationship was obtained between hydrogen peroxide concentration and electrochemical response, with R squared valued approximately 0.9998. We then applied this electrochemical approach. The bacterial samples exposed the molybdenum disulfur. Change in the electrochemical response we're observing depend on the incubation condition and exposure time. This demonstrates the potential electrochemical techniques are complementary tools for investigating hydrogen peroxide related present in biological systems. The future investigation the probes mechanisms. We perform a Florence-based rows. Here we can compare Staphylococcus with the molybdenum disulfur and cleaner in the Florence intensity we observe in the presence of the molybdenum disulfur. The Florence spectrum has also simultaneously increased around 29 nanometers to get peroxidal activity antibacterial. 'Antibacterial' experiment. In the next part, we evaluate where this catalytic behavior could associate with the antibacterial activity. The nanozymes were testing against Staphylococcus aureus and Pseudomonas aeruginosa using controlled hydrogen peroxide conditional. As shown in the graphs, both molybdenum disulfur formulation exhibit significant antibacterial activity. For Staphylococcus aureus, the treatments production very high antibacterial activity after both 3 and 24 hours. A similar behavior observed for Pseudomonas aeruginosa, although some difference between the formulation and concentration we observed. Importantly, control experiments with the hydrogen peroxide In conclusion, with successful synthesis, molybdenum disulfide NANOZYMES with preoxidation activity, the material showed clean catalytic response, and demonstrate significant antibacterial activity against both gram-positive and gram-negative bacterials.